right? So photosynthesis involves two critical stages. We have the light-dependent reaction, which we generally call light reaction, and the light-independent reaction, which we call Calvin cycle, the same that people used to call dark reaction. And again, I've explained that it is not really dark reaction. It's only light independent because it does not depend on light. All right, so photosynthesis consists of light reactions, the photo part, and the Calvin cycle, which is the point where it actually make the sugar itself. The light reaction happen in the thylakoid, and that is what splits water into oxygen. I'm going to use my annotations now. So this is what happens here. Here is the light reaction, which will split water to produce oxygen as a byproduct. So the oxygen that plants breathe out is actually from this water molecule. And so it splits water, providing electrons and proton. So it will split water into oxygen and then hydrogen. Hydrogen here is going to release oxygen as byproduct, reduce the electron acceptor NADP plus to NADPH, simply because the electron released when this water is splitted is going to be captured by NADP plus, and then they will make NADPH, right? Then it's going to generate ATP as well in the course of that process. So the light reaction, number one, produce water oxygen. Number two, reduce NADP plus to NADPH. Number three, produce ATP through photophosphorylation. Phosphorylation simply means addition of phosphate to ADP to phosphorylate it to become ATP, right? But because it requires sunlight, we call it photophosphorylation. And I'll be showing you all of that as we go. Then the Calvin cycle, which is where carbon dioxide, CO2, is going to be reduced to sugar molecule. All right, so the Calvin cycle, here's the Calvin cycle, which is the second part. You see, it makes sugar from carbon dioxide using ATP and the NADPH that was generated from the light reaction. So the way to think about it is that the product of the light reaction is what powers this light-dependent reaction. Does that make sense? These are the product of the light reaction. Light-independent reaction, light-dependent reaction will give rise to ATP. It will give rise to NADPH. And those two are the one that will power the reaction, which will lead to the production of the sugar. So you see, the NADPH is going to come here. It's going to be invested into the cycle. The ATP will be invested into the cycle. And that is what will power the Calvin cycle to produce glucose. So you see, CO2 and NADPH produced from the light reaction will power the production of glucose. All right, so the Calvin cycle, which happen in the stroma, those are tiny details you have to take note of. Take note that the light-dependent reaction, which is the first stage, happens in the thylakoid, and the second stage, which is the Calvin cycle, happens in the stroma. So you want to take note of those details. So it makes sugar from CO2 using ATP and NADPH that was generated from the light reaction. The Calvin cycle begins with carbon fixation. I'll talk about, which is what you see here. So the first thing that happens here is that the glucose, the carbon dioxide, we are going to fix the carbon from the carbon dioxide. And if you think about it, when we were doing cellular respiration, I told you you are burning glucose molecule to release ATP. And every stage, every glucose molecule will give you 6 CO2. If you remember, every glucose molecule, because there's 6 carbon in glucose molecule, you have to breathe out 6 carbon dioxide for you to exhaust a glucose molecule. The same way, for you to build a glucose molecule, plants will also need to take in 6 molecules of CO2 so that they can form 6,6H12O6. Because invariably what is happening here is that we are keeping each carbon here, which is what we call carbon fixation. We are fixing each carbon that come into the cycle. So each CO2 that come into the cycle will fix it. We fix another one, fix another one, fix another one, and we end up having 6 on the long run, which is what we give rise to 6 glucose molecule. That's technically what happened. For you to burn the sugar, you have to release 6 CO2. For you to build the sugar, plant also have to take in 6 CO2. It's just as simple as that. Now, so the carbon cycle begin with carbon fixation, which is what you see here, number 1, which will be incorporating carbon dioxide into an organic molecule to form the glucose, right? It then reduces fixed carbon to carbohydrate by transferring electrons from NADPH to HIF. So the next thing that is going to happen is reduction. What does reduction mean? I told you earlier that CO2 will be reduced, meaning that CO2 will gain electrons, CO2 will gain hydrogen. That's where 6,6H12 is coming from. 6,6H12. It will gain hydrogen, and then it's going to lose oxygen. Because if you think about it, if you have 6 of CO2, now this is going to become C6H12O6. Invariably, this has gained, the glucose here has gained hydrogen because there was no hydrogen here before. Because now here we have 12 hydrogen. Here we have 0 hydrogen. Here we have 6 carbon. We have 6 carbon on the right side as well. On this side we have 12 oxygen. Let me put oxygen in parentheses. We have 12 oxygen here because 6 times 2 make 12. But here we have 6 oxygen. Having 6 oxygen here means that oxygen has been reduced. So removal of oxygen is still oxidation, is still reduction. Oxidation is removal of oxygen, addition of hydrogen, oxidation, then electron also was released, was accepted. So the carbon dioxide was reduced here. That is the reduction process. Do you see how redox reaction is coming in right from chapter 9, coming to chapter 10 now? So that is redox reaction, reduction happening there. Then the last stage, then the last stage, which is regeneration. Regeneration simply means to regenerate what we brought in the first place. I'll be talking about that. To regenerate the system such that the cycle is ready to accept another carbon dioxide coming into the system. So let's go. So that's more like the summary of the whole thing. We are just going to be looking at the breaking down of the process. So photosynthesis involves light and dark reaction, like we said. The chloroplasts use light energy to make sugar by coordinating the 2 stages of photosynthesis, which is what you are seeing here, the light and the Calvin cycle. The light reaction occurs in the thylakoid, like I said. You can see it occurs in the thylakoid. And then the dark reaction in quotes, which is the Calvin cycle or light independent reaction, happen in the stroma. The light reaction is going to generate ATP and NADPH, and also is going to release oxygen to the outside, which is a byproduct. this NADPH and ATP is going to be invested into the carving cycle so that it can power the process there. When they invest, because the electron in air is going to be invested into this cycle, so that it's going to help to reduce carbon dioxide into sugar. Because we need the electron. When carbon dioxide receives electron, it becomes reduced. Now, when that happens, we are going to have NAD plus back. The NAD plus will go back to the light reaction so that it can capture more electrons and become reduced as NADPH again to continue the cycle. Then when ATP is used, then it's going to go back and become ADP so that it can become phosphorylated again and become ATP again. So it go back into the light reaction, into the thylakoid, and the process continue. So the light reaction occurred in the thylakoid and release NADPH and ATP, which are sent to the dark reaction or carving cycle in the stroma, where all energy used will be used to now make carbohydrates or sugar. So again, this is the whole cycle. And so what we'll be doing now is I'll be telling you the influence or the effect of this light that fall to the light reaction, that makes it light reaction. How does chlorophyll react when the light comes into the thylakoid? Don't forget that it is in the thylakoid that we have the chlorophyll. So when the light comes, what impact does it have on thylakoid? How does thylakoid respond to it? And how does it help to split water? And all of those processes is what we'll be looking at as we go. Now, quickly, I'll talk about nature of sunlight real quick. I'll just run through it. This, we call it ray wave. It's showing you different light, red, orange, yellow, green, blue, indigo, and violet, showing you the light and how lights are reflected. So light is an electromagnetic energy. It's also called electromagnetic radiation. They travel in a rhythmic manner, which we call wavelength. So when we say wavelength, we are measuring the distance between crest, and it's simply like this. We are looking at wave. So the crest, this is the crest, which is the peak, and then the distance between them is what we call wavelength. So this is the way light travels. It can range from less than a nanometer gamma to more than a kilometer. The electromagnetic spectrum is the entire range of electromagnetic energy or radiation. Now, what we call visible light is a color that is reflected. So now, all of these colors are usually present in anything that has color. It's just that the one reflected is what we see. The one absorbed are not seen. So these wavelengths, this is the reason I'm looking at this anyway. These wavelengths here are the wavelengths of these colors when they are going to be visible. So that means you expect that you are going to see this color at this wavelength. You expect that you're going to see this color reflected at this wavelength. And yellow is going to be here, but it's going to be absorbed at this wavelength. So for some of you doing this in the lab, you are going to do chromatography, which is going to talk about how the colors are separated. And you will notice that some of you did the experiment in the lab where you pull the pigment from, let's say, spinach or any of these green plants. You put the spinach on chromatography paper and you put it in chromatographic solution. And next thing that you notice is that in few minutes, the colors start to separate. And so you were surprised that just like it was green plant, you put the just pigment from plant, from leaf, you put it on the chromatography solution, and now you are seeing different colors booming out. What it means is that all the colors are present. It's only that green will be visible at that wavelength. because that color will not be absorbed. So like my shirt now, there are multiple colors on it. The only thing is that the color that is absorbed, that is not absorbed, is what is reflected. All right, so these are different wavelengths when the colors are reflected. That's majorly what I need you to see there. So light is made up of photons, which are fixed quantity of energy, which is inversely related to wavelength of light. Shorter wavelengths have more energy. This is what we mean by it's inversely proportional, in that when you have shorter wavelengths, shorter wavelengths, higher energy, lower energy, longer wavelength. So it's inverted, right? If something have lower wavelength, it's going to have higher energy. So this is what you see here. Okay, shorter wavelength, higher energy, higher energy, shorter wavelength. So photosynthetic pigment. So I'm beginning to talk about the pigment now. When we say photosynthetic pigment, the first thing that comes to your mind, of course, is the chlorophyll. The chlorophyll is a green pigment there that attracts sunlight. All right, that's a green pigment that you see inside the thylakoid. And I told you these are the thylakoid. Each of these each of these things that look like pancake is a thylakoid. So this is a thylakoid. This is another thylakoid. This is another thylakoid. And each thylakoid, each stack, we call it granum. And many granum, grana. So if you have multiple granum, we call it grana. So now, if I'm going to say this now, I will say I have 1, 2, 3, 4, 5, 6, 7. I have about 7 grana inside this. All right, inside this chloroplast. The entire thing is chloroplast. Each slice is a thylakoid. Inside the thylakoid is a green pigment, which we call chlorophyll. So pigments are substances that absorb visible light. Different pigments have different wavelengths. So that means they have different light, different colors that they absorb. The wavelengths that are absorbed disappear. The ones that are not absorbed is going to appear. So the ones that are not absorbed, we say that they are reflected. So the color that you see is the color that is not absorbed. That is the color that is reflected. All right. So wavelengths that are not absorbed, wavelengths that are not absorbed are reflected or transmitted. For example, most leaves appear green simply because chlorophyll absorb violet to blue and red light while reflecting and transmitting green. So the reason that plants are green is because the color that is not absorbed in plants is green. Every other color is absorbed, but green is not absorbed. That's the same reason when you now use chromatographic paper, which is color separation techniques, it's going to separate all the colors in green plants, even though they look green, but they have all the colors embedded in them because the colors are absorbed. Let's go. Now, a spectrophotometer is used to measure the pigment, and it sends light through pigment and measure the fraction of the light transmitted at each wavelength. Absorption spectrum is a graph that we use to plot it. Let's go. I'm just punching on that. Okay, cool. So we have 3 kinds of pigment in every chloroplast. We have the chlorophyll A, chlorophyll B, we have the carotenoids. Now, chlorophyll A is actually the major chlorophyll that we talk about when we talk about chlorophyll. So sometimes we even just say chlorophyll, but we mean chlorophyll A. The chlorophyll that is actually involved in the light reaction. So we say that the key light capturing pigment that participates directly in light reaction is chlorophyll A. Chlorophyll B is an accessory pigment. It just supports. Then we have carotenoids, which are a group of accessory pigments as well. All right. So the action spectrum of photosynthesis was first demonstrated in 1883 by Theodor Engelmann. I'm never going to ask you that. You just need to know it as part of the history. He exposed different segments of filamental algae to different wavelengths of light, and he used them to grow some bacteria as a measure of oxygen production. Let's go. All right. So the action of the spectrum photosynthesis is broader than the broad absorption spectrum of chlorophyll. Accessory pigment majorly helps to broaden the spectrum that are used for photosynthesis. I'm just running those because I'm not going to stay on those. Don't worry. So in the last decades, two other forms of chlorophyll were discovered. That's D and F. They also help to absorb higher wavelengths. Cyanobacteria use chlorophyll F in place of chlorophyll A in shaded conditions. You just need to take note of that. Now, I'll focus on what I need to focus on once I get there. Other accessory pigments are called carotenoids. They could be yellow or orange because they absorb violet and blue-green. Carotenoids majorly help to broaden the spectrum as well. Broadening the spectrum simply means it enlarges the scope surface area for photosynthesis to happen, right? Some are also photoprotective. That is, they absorb excessive light that would otherwise damage chlorophyll. So some of these accessory pigments, like I said, they broaden the spectrum for photosynthesis to happen so that they increase the surface area. They also help to absorb excess sunlight so that chlorophyll A will not be, the chlorophyll itself, the chlorophyll A pigment itself will not be damaged because of the intensity of the light. Cool, so we are getting to stuff now. I'm trying to speed up because of time so that we can still capture the things we need to get. Now, when light falls on an object, what happens? It becomes excited. Think about how excited you become when you find out something you have been trying to look for. It's like a knowledge hits you, an understanding hits you, and you're going to become so happy when you find it. Just like you have been trying to get away to a place, and now you find your way there. You're going to be excited. It's the same way pigments, chlorophyll is going to feel when light falls on it. It's like it got an understanding. It's going to be getting excited. So what happened is this. Look at me more than the writings there, please. So what happened here is this. Sunlight falls on the chlorophyll. So think about this as chlorophyll now. Sunlight falls on the chlorophyll, light falls on it. What's going to happen? The chlorophyll is going to become excited. When chlorophyll become excited, what's going to happen? They become oxidized. Write that somewhere in your notes. Chlorophyll, when light falls on the electron, on the chlorophyll is going to be excited and they become oxidized. When they are oxidized, what happens? They lose electrons. So in quotes, the moment the sunlight falls on the chlorophyll become oxidized. Electron is going to jump out. So we say the electron become excited. The electron that was just seated on its own before, but the moment it sees sunlight, is like a child saw candy. And the child will be running all around because he has found candy. So the same way, the moment sunlight comes on the electron become excited as though you just found candy and you are eating candy everywhere. That's the same way. So the electron don't sit again. It becomes excited, jumps out of its hidden place, and now it's everywhere. Does that make sense? Now, so when a pigment molecule absorbs light, one of its electrons goes from a ground state to an excited state and become unstable. You don't give a child candy and the next thing you are seeing is that they are sleeping. They are going to be everywhere. In isolation, excited electron will fall back to the ground, releasing excess energy as if, I don't know if of course, I told you when we're looking at cellular respiration that anytime electron falls, what follows it? Heat follows it. And I told you the heat is like hydrogen ions, then proton. So the same way, when the electron that has been excited, when they are now falling down, what's going to happen? It follows it. It follows it. Now, that is what electron is going to do to chlorophyll. Let's go. So electron will excite that is what photon, sunlight, will do to chlorophyll. Photon sunlight will excite the chlorophyll and make it super active. Now we have two photosystems. Take note of that. Photosystems simply look like electron transport chain. If you still remember electron transport chain from solar respiration, how we talked about how electron moves. So photosystem is close to that. All right. So a photosystem consists of reaction center, number one, surrounded by light harvesting complexes. So take note of that, please. I'll show it to you now. Now, a photosystem, so we call it PS. Now, this is PS. It's not PlayStation. It is photosystem. Now, the photosystem consists of two components. Number one, reaction center. Number two, light harvesting complexes. What does the light harvesting complexes do? They harvest light. They capture light. The reaction center is where you see chlorophyll A itself. Now you will notice that this chlorophyll here have different color from these pigment molecules. So we say that a photosystem consists of reaction center. At the reaction center, what you see at the reaction center is the chlorophyll A itself. Then surrounded by light harvesting complexes. So the reaction center, so we call this, some textbook we call it antenna molecules. All right, like as though they are the one that have the antenna that will receive sunlight. So let me show you what happened now. When sunlight comes here, it's going to fall on this antenna complex or light harvesting complex. It's going to fall on it. Then this will propagate this light that hits this area. So it will send it to this one send it to this, send it to this. What is the intention? To bring it to the reaction center. It will send it to this until it gets to the reaction center. When it gets to the reaction center, that is where we have chlorophyll A. We don't have chlorophyll A in all of these antenna complexes. We have chlorophyll A in the reaction center. So chlorophyll A, once it receives the electron, what's going to happen? It's like a child receives candy. Get excited. If you get excited, electron jumps up. And what is going to happen? This is the point NADP plus is going to come here, primary electron acceptor. Accept the electron, become reduced, and become NADPH, which will be later used in the other reaction. I will get there. I'm just talking about the photosystems now. So a reaction center is an association of proteins holding a special pair of chlorophyll. That's why we call this reaction center. And a primary electron acceptor. So you see, this is the reaction center. It consists the pair of chlorophyll and NADP plus, which is the electron acceptor now in this case. So a light harvesting complex consists of various pigment molecules bound to protein. Light harvesting complex transfer the electron of the photon of the chlorophyll to the chlorophyll A in the reaction center. Does that make sense? So the job of the harvesting systems or antenna molecules or antenna complexes, their job is simply to propagate the photon that they received to where it matters. It's just like as we are here now, Swahili have a question and decide to send it to Chamia first, then decide to send it to Chinyere, then decide to send it to Shegun, then decide to send it to Perpetua, and Perpetua was the one that had the confidence to bring it to me. I am the reaction center which is going to answer the question, but it has to go through different chains until it gets to me. All of the chains that it went through before it gets to me is the antenna complex or the light harvesting complexes. Those people were just harvesting, gathering the questions until it's going to get to me, which is going to answer the question, reaction center. So these chlorophyll A molecules are special because they can transfer an excited electron to a different molecule. That's what is characterizing chlorophyll A. I'll talk about that together. So I know I'm running quite fast, but I believe I'm not too fast for you. Now, the primary electron acceptor is here. A primary electron acceptor in the reaction center will accept the electron that was excited from the chlorophyll A and become reduced to become NADPH, right? So solar power transfer of an electron from a chlorophyll A molecule to the primary electron acceptor is the first step of the light reaction. So here is the first step of light reaction when the electron is released here and the electron reduces the electron acceptor to NADPH in quote. Let's go. So there are two types of photosystem, like I said. There's photosystem 1 and 2. However, we call it the one that comes first is photosystem 2 and photosystem 1. Let me tell you why. It is not PS1 and PS2. It is PS2 and PS1. The reason is 2 first is because 2 was the one that was discovered first. So when that was discovered, PS2, then the second one discovered, PS1. That's why the numbers are alternated. You expect that it should be PS1 first, then PS2 first. No, it is PS2 first, then PS1 first. Because this was the photosystem that was discovered first. Then the second was later discovered, which is 1. All right. Now, so there are two types of photosystems in the thallicoid membrane. I told you the light reaction happened in the thallicoid membrane, and this is the light reaction. It happened in thallicoid. So that means there are two photosystems there. Now you see they are numbered in their order of discovery. Okay, that's why it is 2, then 1. Now, another way to describe photosystem 2 is to call it P680. And photosystem 1 is P700. Now, so you see here P680, P700, which is photosystem 2. Both of them have the same orientation. There's a reaction center, which is where you see chlorophyll A, and there is the antenna complex or light harvesting complexes, which is what propagate the sunlight to get to the reaction center. Sunlight doesn't hit the reaction center directly. It comes, they will capture it, then propagate it until it gets to the reaction center. Now, so P680, because its reaction center chlorophyll is best at absorbing light within the 680 nanometer, and then P700 because it's able to absorb light within this wavelength. That is why we call it that number. Now, I will run through what I've said again for you to emphasize it, and then I will stop this video and record and start another. So now look at it. Light comes in through the antenna complexes or light harvesting complexes, transfer the light, the photon keeps traveling until it gets to reaction center. When the photon gets to the reaction center, what happens? The photon will get the reaction center to become excited. When the reaction center is excited, what happens? Is it going to be seated? No, the electron is going to jump up. When the electron jumps up, you have the electrons here. What's going to happen? The electron will be accepted by the primary electron acceptor, and then it's going to be reduced once it accepts the electrons. I'm coming to show you that, don't worry. because it's processed, there are two photosystems. So I'll show you what happens there. So the electron will be accepted here. Now, think about it. Let's think about something since you understand redox reaction now. This electron was removed from the chlorophyll reaction center, the chlorophyll A that is here. That means this chlorophyll has been oxidized because it has lost an electron. It has lost the electron here. Two electrons were lost, actually. Now, if it loses this electron, what do you think is going to happen? It's going to stay in this excited state and be oxidized and not become stable back? No, it has to get back to its natural state. So the electron has been lost, accepted by this primary electron acceptor. It's going to travel through this chain, which I'm still going to talk about. So something has to happen to take care of this state, this chlorophyll, so that it's not remaining unstable. To attend to the stability, then the light is going to split the water molecule. The electron is going to split the water molecule that is here, split it into oxygen, and then anytime you see H, don't forget, hydrogen is equal to H plus E minus. This is hydrogen. So even though hydrogen molecule was splitted into O2 plus hydrogen. So if you have 2 here, we'd rather have 2 here again and balance this. Don't worry, this is just to balance the equation. Now, so that means we have 2 of these. If you have 2 of these, it means you have 2 of this. Because H simply means H plus and E minus. So if you have 2 of that, it means 2 times this. And that's what you see here. Then these are the 2 electrons. You know what they did? They came back to replace the electron that was lost here so that this guy can become stable and be able to sleep. Because now this is like somebody that has been fired up and cannot sit to have rest. It's unstable. So to maintain that stability, water molecule splitted and replace the electron that was lost. Now, let me ask you a question. Chlorophyll A at the reaction center in PS2 lost electron when it received photons, light, and became excited. It lost electron. How was that electron restored back? The electron was restored by when water molecule was splitted. That was actually the reason for splitting water molecule in this stage. It is so that it can replace the electron that was lost at the reaction center of PS2. This electron that has been lost is going to travel through this chain, just like electron transport chain. But the electron, this guy has to be stable. It can't remain unstable. So that was what necessitated the splitting of water molecule, so that it can help to replace this electron that was lost here. So next thing, this electron is going to travel through these protein complexes. It travels through protein complexes, and you know what's going to happen? When it travels through here, photon light also is going to hit this guy. Then it's going to be propagated through here, and then what's going to happen? It's going to release electron. This guy is going to release electron as well and become excited. When this is excited, it has also lost electron. It became oxidized. It also needs to be stable. So what's going to happen? The electron that was traveling from here is going to come here and replace the electron that was lost here. So that will take care of the stability. Now, let me ask you a question now. The chlorophyll A reaction center in PS1 lost electron because of its exposure to sunlight as well. How did it attain stability back? The electron lost in PS2 helped to replace the electron lost by PS1. What replaced the electron lost by PS2? Water molecule released electron, and those electrons replaced the electron in PS2. So a chain. PS2 helped PS1. Water molecule helped PS2. So the electron lost here, we go through this electron, go through ferrodioxazine, goes through that way. Then you see that's the electron that will now be accepted by NADPH, NADP plus, and reduce it to NADPH. So this is how NADPH is formed. I already showed you how oxygen is formed, and ATP is formed in the process by coupling ADP plus phosphorate. So that is what happened in the light reaction. So in this light reaction now, we've talked about how NADPH forms, how oxygen molecule forms, and ATP forms. I'll stop there and make it the next one, another video.